Hello and welcome back to my channel, Explaining Biology Clearly. Today we're going to talk about polysaccharides, and the polysaccharide that we're going to focus on is starch. I do have videos on disaccharides and monosaccharides, so do check back on my channel to find out more about those. So polysaccharides, what are they? So polysaccharides, first of all, are a type of carbohydrate. And a carbohydrate is a biological molecule that you find in living things that is made of only 3 atoms, and they are carbon, hydrogen, and oxygen. Now, polysaccharides are the polymers. So polymer, the word polymer means many units joined together, many monomers joined together. So polysaccharides are made up of lots of monosaccharides. So they are made of multiple monosaccharides joined together. One type of polysaccharide is starch, and that's what we'll look at today. So starch is found in plants. It's a storage molecule. And what it stores is, or you can either say glucose, or you could say energy. Starch is made up of glucose, hence why it stores glucose, and glucose is used in respiration to release energy, hence why you could also say that is a storage of energy. So there are 2 types of starch, one of which is amylose, and that's this blue picture here. And the other type of starch is this orange picture down here, and this is a myelopectin. So first of all, let's have a look at the blue diagram, amylose. So as I said, it's a polymer, so it's made of lots of monomers, and the specific monomer is a monosaccharide. And the monosaccharide that makes up amylose is alpha glucose. So it's made of lots of alpha glucoses joined together, and they're joined together by this bond here, this oxygen, and this bond is called a carbon 1 to carbon 4 glycosidic bond. The carbon 1 to carbon 4 describes the position of the bond. So this is the position of the carbon 4, and here is the position of the carbon 1. So it is the bond is between the carbon 1 and the carbon 4 of the molecule. Carbon 1 there, that's the carbon 1, and the carbon 4. So it's a carbon 1 to carbon 4 glycosidic bond. Again, carbon 1, carbon 4, carbon 1, carbon 4. Once that amylose is produced, we have a straight chain, and that straight chain doesn't remain like that. Because of hydrogen bonding, it folds into a helix. So it takes on this structure here, and that is held in place by hydrogen bonds, H bonds, hydrogen bonds. These are weak forces of attraction between negatively charged oxygen and positively charged hydrogen that is sticking out from these alpha glucoses. Okay, so that's amylose. So let's take a look at a myelopectin. Now, a myelopectin has a very similar structure along this straight chain. So if you look at this straight chain down there, what you'll see again is alpha glucose molecules joined together by carbon 1 to carbon 4 glycosidic bonds. So the main chain, we call this the main chain, the straight chain, is the same as in amylose. However, there's one main difference, and that is these side branches. So a myelopectin has side branches. It's quite highly branched. I've only drawn one branch, but it will have multiple branches off this main chain. and the side branches are joined to the main chain by carbon 1 to carbon 6 glycosidic bonds. So up here is the carbon 6 and this is the carbon 1 position. So the bond is between the carbon 1 and the carbon 6. And that is just the bond that joins the side branch to the main chain. The side branch itself, again, has carbon 1 to carbon 4 bonds. So it's joined in the same way as this main chain. So again, here we have a carbon, actually I've drawn a carbon 6 to carbon 1 glycosidic bond there, because we have a side branch off of a side branch. Okay, so that's a basic description of their structure. Now, one thing you may be asked about in a post-16 biology question is linking the structure of these molecules to their function. Structure and function. So anything that mentions explaining structure and function, these are some of the things that you want to say. So if you think about their function, we mentioned it earlier, their function is in glucose storage or energy storage. So in order to store that successfully, it's important that they are insoluble. They don't dissolve in the cytoplasm of the plant cell. So this structure that they have makes them insoluble, and being insoluble means that the molecules don't affect the water potential of the cell. That symbol stands for water potential. So they don't cause the cell to lose or gain water by osmosis. They don't dissolve, and therefore they don't change the solute potential of the cell or the water potential of the cell. So another answer that you might give is that they're easily hydrolyzed, and that we will explain in just a moment. They're easily hydrolyzed, and they're easily hydrolyzed because there are lots of ends, and therefore glucose can be released from the molecule. Now, what do we mean by hydrolyzed? In order to break this glycosidic bond, you need to add water, add a molecule of water. Breaking by adding water is a hydrolysis reaction. So we say that it can be easily hydrolyzed. These bonds can be easily hydrolyzed, and that then releases this glucose molecule, so it can be broken down in respiration. I think of a myelopectin in particular. There's the main chain, and you've got these side branches coming off like this. If you looked at it from a distance, it may look something like this. You can see, hopefully, that you've got these ends where enzymes can attach and break off glucose so that it can be hydrolyzed. And then the final answer that you might like to say is that they are quite compact. Think of amylose in this case. By coiling up that straight chain, you're squeezing lots of glucose into a small space. So lots of glucose stored in a small space. So that may also fit with a question about the advantages of starch as a storage molecule for glucose, as well as a question on describing and explaining how their structure and function are related, or describing and explaining how their structure aids their function, anything along those lines. I hope you've found this video helpful. Don't forget to like and subscribe to my channel, and check out any more videos on carbohydrates, monosaccharides, disaccharides, and other videos that I have on the channel. Thanks very much.